right. Hope you all had a lovely break and you got some good socializing and networking in there. So on to the next talk. We've got two presentations from the Central Hub. So first on is Josh Quick, and Josh is going to talk to us about the sequencing journey. Josh, can you hear us? Can you see us? Can we hear you?
Yeah, I can hear you. Can you hear me?
Amazing.
Yes, we can.
Great.
Okay, great. Hi, everyone. Somewhat ironically, I'm presenting this from my spare bedroom because one of my friend's kids has got COVID. So I'm not able to be with the Birmingham and Central group, unfortunately. But I'm just going to share my screen. Hey, can you see the slide?
Yeah, brilliant.
Great. So my role within COG-UK was chairing the sequencing working group, and it was a really great group of people, and we did I think a great job of sort of managing the protocols in use within COG-UK. I'm going to talk a bit about the protocol. So there's lots of considerations to take into account when you're trying to identify a suitable protocol for what you want to do. And we've been working on this for quite a while, since working on the Ebola and subsequent Zika outbreaks, developing really robust, easy to do sequencing protocols. And obviously this was the primary aim of those efforts was sort of field considerations that you'd consider a sort of time to answer, cost and ease of use, risk of contamination, portability and scalability, I suppose. So and obviously there's different modalities of sequencing, if you like, the targeted and versus untargeted. If you're looking at the untargeted methods, such as sort of direct RNA or RNA-seq, those sort of RNA metagenomics, you can identify or pick up things that you're not necessarily certain, but in amplicons and qRT-PCR, which are limited on what you can actually detect, but then they tend to have higher sensitivity. So that's sort of the thing that you're trying to find the sort of best protocol really that suits everything. And this graph here shows the viral loads, or the CT values for the samples that we collected during Zika, and you can see that typically Zika has a high CT value. These have a modal CT of 36. So when we're developing
Sorry to interrupt, Josh. You're breaking up a little bit at this end.
When we're developing the sequencing protocol for Zika,
so Josh,
yeah,
it might help if you turned off your video, so you're breaking up a little bit. Sorry.
Is that it better?
Yeah, that's great. Thanks.
Can you still? You can't see the slides now, presumably.
No, we can see the slides.
Okay. Yeah. So for the Zika samples, the modal CT value was pretty high, CT 36. So the protocol that we developed was really by necessity one that had high sensitivity, and for that reason we chose the amplicon sequencing. It's worth noting here that not every sample that becomes positive by qRT-PCR can be sequenced, because of this difference between targeting just a single region or multiple regions of the genome versus trying to recover the whole thing. Obviously, a partial genome could still be recovered from a high CT positive. Okay, so this is the concept of a viral sequencing method scalable to thousands of samples. So amplicon sequencing is cheap, and it is easy to scale. It works really well with clinical samples and isn't so dependent on RNA quality. We tend to target shorter amplicons around 400 base pairs, although some people have shown that longer ones, longer amplicons work well. You can recover genomes or partial genomes in the range of 30 to 35. And the way that we do this with the Arctic method is using multiplex PCR followed by barcoding and nanopore sequencing. Although at an amplicon length of 400, it is compatible with Illumina as well. And then the reason we choose nanopore sequencing is because it's real time and generates high quality consensus genomes using a reference-based consensus approach. So this was our original paper describing the multiplex PCR method in 2017. And fast forward to 2020, and this was the GenBank record for the first what would come to be known as SARS-CoV-2 genome, which was published by Fudan University in January 17th. This version, January 17th, 2020, and me and Nick were talking about this the other day. I remember him. Yeah, I think we should design off that original genome. It came out on January the 22nd. And then we'd been previously working on the North Kivu outbreak of Ebola in the DRC with Ian and Nick and a few of the other collaborators, and we just forked the protocol, made the version 1 of the NCOV 2019 protocol and put it up on protocols.io, which is our standard way of sharing protocols online. Little did we know that it would become, massively popular. This software that me and Andrew Smith in Birmingham developed called Primal Scheme. You basically upload the genome to the website, and then it just processes it and finds the designs the primer scheme off the genomes provided. And then once you've got the primer scheme, you typically order the primers and then do a multiplex PCR in 2 pools from cDNA, combine them, and then in the Arctic method you do end repair, DA tailing, and native barcode ligation, followed by sequencing adapter ligation. And the reason that we do this is because we find that it gives firstly it gives full length amplicon products, and it gives very high rates of correct demultiplexing on nanopore, which is why we stick with this as opposed to sort of alternatives. And then this is moving into some work that this was done to but also within the up to version 2 and 3 of the lab protocol, which we called low cost, and that was through a combination of adding in primers to address the amplicon 64 issue, which was by reducing the annealing temperature to 63, adding in alts. We made changes to the end repair and ligation protocol, which actually improved the barcoding efficiency from about 40 to 75%, which meant you could use more usable data. And ONT brought out the 96 barcode native barcoding kit. And so combining all these, you could get cost reduction down to about £10 per sample. Really, Itakawa et al. Released a paper where they actually sequenced the dimers from the Arctic PCR and identified that there were some reproducible dimer products which would form. And specifically, these were heterodimer products which had exact 3' matches with other primers in the pool or themselves. And I thought this was really interesting because none of these dimers have a TM of predicted TM of anything like what somewhere 30 degrees. We're running the PCR at 65 degrees, so that's sort of an interesting discovery. And it's clearly due to the really long annealing temperatures that we use in the PCR, which allow the multiplexing to work. It means that these dimers, which we weren't looking for before, can cause a problem with forming dimers. So that was something which we're able to roll into the subsequent versions of Primal Scheme. This is the COG channel from protocols.io, showing the sort of the main three initial protocols which were released, the sort of high throughput protocols, if you like. There was the 96 version of the tail probe based target enrichment. So we sort of moved into the sort of high throughput phase of the sequencing protocols. And this is a quick outline. I haven't got much time, so I'm not going to go through all these, even though I'm sure you'd love to, of the different sort of protocols that are in use. So there's a tailed protocol at Sanger, which uses a normal multiplex PCR followed by a 4' PCR using the tailed primers to generate these. There's a group of Nextera type protocols which include COVID-seq from Illumina, Corona Hit from the Quadram Institute, and Mini-XT from the Northern Ireland group. And all of those use sort of miniaturized versions of Nextera XT on Arctic amplicons. And then the probe-based method from Oxford, obviously using RNA-seq followed by bait capture. And then you've got right at the sort of near the end of the COG sort of sequencing, there was an RC-PCR method that was actually developed by two people at the Salisbury NHS Trust, but was commercialized by a company called Nijmegen in the Netherlands called RC-PCR, where you actually use heterodimers to create functional primers. So that's quite interesting. And that's definitely very scalable. This is a slide from protocols.io showing the release of the sort of main class of protocols, the initial PCR and Arctic protocol, followed by sort of high throughput 96 and 384 well protocols, first wastewater, direct, etc. You can see that the number of views and the exposure of these protocols is absolutely massive. In fact, the Arctic protocols had 175,000 views, and if you look at all the COG protocols together, it's over nearly 250,000 views. So, so that's an extremely good way of getting protocols out there, I think. In addition to that, we couldn't, you know, the protocol was kitified by New England Biolabs into the NebNext Arctic protocol, and IDT. We had to engage with some commercial oligo providers, because initially I was making up, or Nat and my group was making up primer pools, and we're sending them out by DHL just individually, and it was just too difficult to keep up with the demand. So I think it did allow people to get started quite quickly, obviously with the shortages of oligos that there were, but we needed to get a commercial partner in, and IDT luckily were able to do this really quickly. That was especially important when DHL services just stopped running at the university, and I had to physically drive them over to Prologis Park in Minworth if I wanted to post anything. So I'm really grateful to IDT for coming, you know, being able to supply the pools in vast quantities worldwide. But obviously they did hike the price, so we've now managed to get Sigma on board as well, to get the price back down again. So that's really good news, but that's only just happened recently. Yeah, so another thing that we've done within the working group was to monitor the primer. Some people call it erosion, if you like, the primers that's failed to work due to new variants. And if you look at this, you know, this plot here, you can see all of the mutations in the common variants of concern, and they sort of cluster around the spike protein around 20 to 23,000. And what we did for V4 was, you can see that there's these failures that are occurring in the V3. So we had to create reference genomes or pseudo genomes for these VOCs and produce a new primer scheme, and that was V4, which was rolled out, tested within many sites in the working group, and that was able to recover the full spike protein for Delta and Beta, and that was successful. We had many other smaller projects within the working group. We had a false positive investigation group where we were attempting to identify diagnostic false positives using genome sequencing. We had a protocol. We had an investigation looking at the use of accugenomic spiking controls for the Illumina sequencing run. We investigated and fixed the Amplicon 64 dropout in the group. Like I said, temperature. We looked at hologenetic experiments and knowledge that each site was individually getting, as well as the new kits available, like the 96 barcode kit and the Luna script. Run quality tracking using setting up a COG Minotaur server, and then more recently the Amplicon 74 dropout. So that was really quick, but I've only got 10 minutes. So thank you for listening. And this is all the acknowledgements for the RTIC network, and then this is the whole DL for the COG-UK sequencing working group. So thanks very much.
Right, thank you very much, Josh, and thank you for not giving up in the face of adversity, whether it be it Zoom or DHL. So now we're traveling over to Birmingham. Hello, sunny Birmingham. No, that's a different show. So now from one person who had to sort of take matters into their own hands and drive to David Bonsall, who actually drove over on Christmas Eve. All the way to Cambridge. So David, can you hear us? Can you see us? Can we hear you? You're on mute.
Hi, yeah, we can hear you. Can you hear me?
We can hear you.
Oh,
Thank you.
we can't.
And we can see the video on your computer.
We can hear you, Dave.
Can you hear me?
Sit down.
So we can't hear them. Is that either? Can you hear me, Josh? Josh, say something.
David, we can hear
Somebody
you.
say something. Okay, that's good. So we've got a very complicated audio AV system that Nick has wired up literally on me. So you can see everything that I can see.
Okay, so we're going to be watching you. Perfect.
So, and apparently this is the best way of doing it, because Nick says so.
Perfect. David, can I just, before you start, at the end of your talk, if you go to the bathroom, do remember to take it off.
Right, yeah, I will, thank you. Yeah, so that's true. So I did drive to Cambridge on Christmas Eve, which was before I did a shift actually on the John Waring ward as the registrar on call for the Christmas pandemic doctor for Oxford. With about 40 kilograms of dry ice and the world's most precious samples, the vaccine samples and the ONS samples, because the sequencer broke just as the alpha wave was taking off, and I had the windows down when I was driving, so I didn't asphyxiate. But can you actually can you see my screen? That was no, you probably can't yet. Sure. Okay, so you should be able to see that.
That's perfect. Thanks, David.
Okay, all right. So I've done literally the opposite of what I was asked to do, which is to sort of focus more on the future and sort of where we're going with the approach that we've taken. So I think we're the only sort of high throughput sequencing lab, I think internationally, that's used sort of non-Amplicon non-PCR virus-specific PCR approaches to sequence. And I want to give some of the context as to why we sort of were so belligerent and thought about that. And that's because before we actually started working with COG, we'd just been awarded funding through the Gates Foundation to think about systems, high throughput systems for genomics integrated with diagnostics that we would put in the low income settings. And the core of that work actually is to provide diagnostics and molecular epidemiology for endemic diseases, especially HIV, which is the disease that we've worked on the most historically. But you know, also thinking about how such systems could be used for pandemic prevention, outbreak response, and I think it's an important point to make that, you know, pandemic response is not the same as pandemic prevention, and the tools that we need to use that are most useful and most valuable are not necessarily the same tools. We actually approached the pandemic as a group, working with Christoph Faeser, from quite a strange place. We've been working on the sort of the modelling of the epidemic, and we're looking at the dynamics of transmission, and came up with the idea that that we could do sort of a better job at contact tracing if we use smartphones, and that led to a whole piece of work that you're no doubt familiar with, that you've probably got on your phones. But it sort of framed our thinking in terms of the importance of speed and the importance of turnaround time for both diagnostics, so you can do your contact tracing, but in the context of a new outbreak and a new pandemic, you know, you need that information faster to initiate responses that could potentially contain outbreaks. And so I like to think of the problem moving forward as sort of the sorts of technologies and systems you would have liked in Wuhan at the very beginning of the outbreak, that could have guided more effective responses and that could have prevented pandemic. So as I mentioned, we approach sequencing with non-amplicon approaches with a protocol we call VSeq that I developed with Maria Teresa when she was a postdoc working with us, and we have used that to process around 30,000 SARS-CoV-2 samples, most of them COG, not all of them. In fact, I'll point my camera, that's George there, that's our technician who has had his hands on literally every one of those samples. It is a high throughput method. But the general strategic direction that we sort of are moving in and have been moving in is to think about all these domains. We want it sort of integrated more with diagnostics. We want it fast. We want to think about, you know, the networks and build on those networks that have been built through infrastructures like COG. We want the platforms to be scalable, not just high throughput, but to sort of cope with low throughput and high throughput, and to be cost effective and appealing, accessible. But importantly, it needs to be broad, and it needs to be able to handle those pathogens that you don't know, as well as those that you do at the point that they emerge. And as an appraisal of the sort of pandemic response that has been a very ineffective pandemic response, and sort of where we need to think about what we need to think about in terms of converting this to a system that matches sort of requirements for prevention. This is a schematic, and I'm sure people are going to challenge me on some of the sort of averages that I've sort of pulled out here. But you know, you've got to consider the end-to-end turnaround times. I mean, this came out of the recent inquiry report. You know, it's okay focusing on your bit, but ultimately the success is in the completeness of the system and the end-to-end turnaround time, and really the time it takes to get that sequencing result after you've considered the time it takes for somebody to consider whether or not they even want to do a PCR test in the first place is currently sort of over a week. And we're thinking about ways that we can sort of reduce the latency in that sort of engineering system. But a real an important thing is that if you really want these systems to work in for your pandemic prevention, is that, you know, they need to be pathogen agnostic. There's no point having to need to know the genome of the organism that's causing your outbreak in order to do anything about it and run your diagnostics. So the protocol that we had in operation that we've used for diverse RNA viruses like HIV and hepatitis C is this protocol VSeq. It's a bait enrichment protocol that starts by extracting total nucleic acid from samples. We prepare our libraries, and then we enrich with panels of probes. The reason that we were fixed on that before we started the pandemic, because as I said, we work on diverse RNA pathogens, and we very much care about sort of unbiased representation of you know, genomes that can differ by sort of 30%, even within a population in specific areas of the genome for HIV, and it's very challenging. It has been tried to sort of use amplicon methods for that. So that's sort of really where we were, and then a recent sort of development is to think about how you can then integrate sort of diagnostic processes within that pipeline. And so a process that we've actually come up with is to sort of construct your libraries, and then during the library preparation, perform your quantitative PCR-like diagnostics for viral load quantification of specific pathogens, or multiplexed PCRs for multiple pathogens, or for specific genotypes and mutations. And that data has two purposes. One is to so you can return that information very quickly to your to individuals as clinical data, and the other is that you can use that that information to optimize sort of downstream enrichment sequencing. So we demonstrated that this actually works. This process works. This is just to say sort of where we've sort of determined the genotype by whole genome sequencing using VSeq. You get a few more when you genotype with the reflex assay-like methods. Those are the qPCR tests provided by Thermo on the RNA. When we perform that assay integrated within our library preparation, you get the same result. And the quantification of those pathogens using that method integrated with the library preparation correlates with the clinical viral load of the sample. Actually, this on the left is sort of data from SARS-CoV-2. This on the right is data from HIV, which makes the point that it's a system, a single system that can sort of unify sort of processing of multiple samples from diverse sample types, plasma, respiratory samples. So there are advantages to generating the nucleic acids as the first step of doing this, because it produces that single system for diverse samples and pathogens, and it generates that amplifiable template for diverse applications. If you remember when sensitivity dropped off from the Sanger Institute because we started doing reflex testing, there was less material available for the sequencing. So this sort of solves that problem because you generate your sequencing libraries first, and that's an amplifiable template that you can sort of use in multiple sort of and it's also sort of your fastest feasible route for the discovery of new pathogens. So this was a grant that we sort of have and is in motion in Zambia at the moment. We're creating sort of high throughput hub and spoke model sequencing platform with I can give some of the sort of details of sort of the hub. This is a sort of a mock-up of the lab that we have in design at the moment that will implement the sort of VSeq protocol in Zambia. The whole thing costs about $1.3 million with a high throughput sequencer. That's if you put a NovaSeq in it, and it has a sort of throughput of about 1,500 samples a day. And this is tested. This is what sort of George was doing actually in the heat of the pandemic for generating libraries in a single day. So in theory, you can then turn around your clinical diagnostics from that 1,500 samples within that day, and then select sort of those samples that you go on to sequence. And we think that sort of with a NovaSeq, you can do somewhere between 4,500 to 6,000 sequences a week. So this is how it might work for sort of HIV. You could imagine that you've got individuals that come forward and they want to know their viremia for HIV. They want to know if their treatment's working. They want to know if they've got any drug resistance mutations, and they want to know if they've got any other pathogens. You can produce your libraries from the plasma that come in, and you can identify within a few hours of receiving the sample that they've got HIV. You can tell them what their HIV viral load is. You can tell them if they've got hepatitis B. Tell them what their hepatitis B viral load is, and then you can return that information to them same day, and then select those samples that you want to send on for sequencing, and that turns into your sort of molecular epidemiology data sets that you need for public health and guided interventions. So it's a system that sort of brings together the diagnostics with the molecular epidemiology. I'll just skip over that because I think I might be sort of going over time. Well, actually, just to point out that, that comparably this method, as we sort of cost it and compare it to sort of commercial viral load systems for HIV, it's comparably similar. So it's within the sort of range. That's the sort of cost for getting to the point of your sort of qualitative diagnostic of sort of viremia and quantification. And then your sequencing costs with this are sort of an additional £14 compared to the sort of $73 that people charge for resistance testing in low-income settings, and sort of similar throughput. But this is where the system really comes into its own for sort of pandemic prevention. You imagine that you get a cluster of cases with sort of symptoms of a viral hemorrhagic fever somewhere in the northwestern province in Zambia. There's a cluster of cases, so you suspect viral hemorrhagic fever, so you generate your libraries and you perform your sort of multiplex qPCR during the library preparation to look for the likeliest culprits. But you don't get one, you don't get a diagnosis. But at that point, you've already generated your sequencing libraries. So you are ready at the point that you have suspicion, but you have a negative diagnosis for a likely pathogen, you are ready at that point to put that sample directly onto a sequencer and generate your metagenomic sequencing output and discover a new pathogen. Now, and at that point, of course, you've then got a system that could also serve as sort of temporary high throughput diagnostics for that pathogen. So another sort of direction that we've taken the lab in during the pandemic is to consider accurate long read sequencing. And so VSeq generates total nucleic acid libraries that are sort of representation of the total nucleic acids in the sample. Illumina, if you sequence on Illumina, you only see the shorter fragments. If you sequence exactly the same libraries, you get much longer reads. PacBio generates fewer reads, but they're longer, so you need fewer of them in order to generate whole genomes. You know, in some contexts, actually, the sensitivity of a PacBio run that only generates 2 million reads, you can get sort of the same sort of sensitivities as a sort of a NovaSeq run. And actually, the advantage of sort of this for the types of analyses we like to do, and this is HIV data, this is using the within-host phylogenetic information and the between-host phylogenetic information. So looking at the quasi-species of multiple hosts that are linked and drawing these phylogenetic trees where you can actually see how the phylogenetic, how the sort of quasi-species phylogenetic structure originates from the phylogenetics of the recipient. So in a cohort of known epidemiologically linked pairs, and there's about 100 and there's about 90 epidemiological pairs here. We can use this information with a pipeline called Phyloscanner that Matthew Hall and colleagues developed to actually infer the direction of transmission. And you can see that wherever you have a red dot below our threshold for calling transmission and a green dot for calling transmission above the line with PacBio, that's an improvement in our ability. And so we feel that in some contexts, our sensitivity to detect transmission of viruses with accurate long read sequences is about twofold using this. So, I mean, that's just a whistle-stop tour of sort of some of the developments that we've sort of pursued and the direction that we're going in. Thank you very much. I'd like to thank everybody here and for the opportunity to talk. Back in Oxford, the Oxford Genomics Centre, the Wellcome Centre, and Katrina, who's with us today, and George and Matthew, who've been absolutely instrumental, and our collaborators in Africa who are helping us get this lab off the ground. So thank you very much.
David, thank you very much. That was excellent.